Good afternoon, everyone. I'm Liz Fernandez with the Professional Development Services Branch, and I'd also like to welcome you to today's webinar. Our speaker today is Dr. Keith Bailey. Dr. Bailey is a clinical associate professor and comparative pathologist at the University of Illinois College of Veterinary Medicine. He is the immediate past president of the American Association of Veterinary Laboratory Diagnosticians and served as the director of the Oklahoma Animal Disease Diagnostic Laboratory from 2014 through 2019. In addition to veterinary diagnostic medicine, Dr. Bailey worked for 7 years in the biopharmaceutical industry. His current research interests include comparative and computational pathology, with particular emphasis in breast cancer research, and his veterinary research includes evaluation of an African swine fever virus model in pigs. He is also currently co-editing a book on the tactical sciences for biosecurity of animal and plant systems. Dr. Bailey is a diplomat of the American College of Veterinary Pathologists. And with that, I'm going to turn the webinar over to Dr. Bailey.
Thank you, Liz. I appreciate that introduction. It's certainly a pleasure to get to share some of the results that we have from a study that completed last month in Oklahoma. I do want to acknowledge that this was a big effort. The study was part of a multi-institutional and multi-agency partnership. And I'd like to recognize some of the contributing members. Envirotech Engineering and Consulting, Jeremy Seeger, who was previously at the Oklahoma Department of Ag, Food and Forestry, the National Pork Board, the Oklahoma Department of Ag, Food and Forestry, the Oklahoma Pork Council, specifically Roy Lee, Oklahoma State University, several members of the team there, myself, the co-PI, Fernando Bowerman, USDA APHIS, who was a critical component in planning and getting this study underway, and the Virginia Department of Environmental Quality, Gary Forey. This audience is keenly aware of the projected high mortality if African swine fever gets into the United States swine industry, and is also keenly aware of the complexities that companies will face with the safe disposition of animals that die on infected premises. And so for us, what we were trying to do is evaluate an immediate, safe, and effective option for carcass disposal that takes into consideration some very important factors such as public health risk, the biosecurity risk, animal welfare, environmental regulations, resource availability, public perception, and farm sustainability. So our hypothesis going into this study was that above-ground burial is an effective and biosafe option for the on-site swine carcass disposal and virus inactivation. So we had to select a surrogate virus for this study, and given that Asparviridae family only has one genus, which is Asparvirus, and African swine fever is the only virus in that family, we didn't have a super close relative, and so we decided to select a surrogate virus that had a very similar linear genome and had other replication cycle components that were very similar to African swine fever. So we selected Swine Pox Virus. The other issue that we had to consider is that by doing a study in the field, we needed to select a virus that we could safely place in the field and still be able to contain, and that was reviewed by the Oklahoma State University Institutional Biosafety Office. And one part of that is if we put virus in the field, then we are obligated to retrieve any contaminated materials that go in the field so that we can bring those back into the laboratory and autoclave those. And so we selected swine pox virus. Pigs are the only natural host, and humans are not infected. It is virtually ubiquitous in swine populations. We chose the femur as our sole site of virus inoculation because during the decomposition study, you're not going to be able to retrieve all of the bodily remains, and the bone tends to be the last surviving tissue that could be identified at the end of the 12-month study period. So the bone was selected, the femur was selected for this, and we'll refer to swine pox as our surrogate virus. So we had 4 main study goals: to evaluate the effectiveness and suitability of above-ground burial for the mass disposal of swine carcasses on the farm, to evaluate the viability of a surrogate virus in the femoral bone marrow over the 12-month period, to assess the local environmental impact of this process, and to characterize insect activity at the burial site. So our study design was pretty simple. So we acquired, with the help of the National Pork Board, 100 cull sows, humanely euthanized those sows, and then inoculated each femur with a surrogate virus, swine pox virus. We characterized the carcass decompositions under the conditions of above-ground burial for the 12-month period. During the study, we recovered both femurs from 5 sows, and we did this at intervals at the end of each week, 1 through 4, and at the end of each month, 2 through 12. We trapped and identified insects at the burial site either weekly or biweekly, and for the latter, it was based on wintertime, so we did it less frequently in the wintertime. We utilized cameras so that we could document any potential soil disturbances or predation at the burial site. We assessed the environmental impact of the study, and then ultimately at the end, we will ensure land reclamation. And I say we, this is one of those group efforts where it's very light on me per se, but it's pretty heavy on the rest of the group. And so I want to acknowledge that up front because some people carry a lot of the weight of this. Again, this was a 12-month study. It started in September of 2019 and it concluded last month. So I'm sharing a lot of preliminary data that was either acquired during the interim period and has been QC'd, or some of it is still pretty fresh and it's subject to final a detailed analysis and statistical analysis. So one of the first questions that people often have when I mention above-ground burial is, what is above-ground burial? I had the same question when this project was presented to me, because in my mind I was thinking that you would simply just put carcasses on the ground, but that's not it. So it's really a shallow burial, and this is an illustration that was put together by Gary. And if you look at the cartoon, the dark body in the middle is actually an animal carcass. So you dig a trench, a shallow trench. You put a carbon source, which is typically wood shavings or straw. Place the carcasses on the carbon source, and then the soil that you dug out of the trench you put on top of the carcass. And then ideally plant some vegetation on top of that to minimize cracking and fissuring. So that's the concept of above-ground burial. So where did we place the study? It was placed at Oklahoma State University, and you can see in the little outline of Oklahoma there, that's about the location of Stillwater, Oklahoma, so north central. And for those familiar with the swine industry in Oklahoma, which there are a lot of pigs in Oklahoma, the panhandle, those 3 counties that make up the panhandle, is virtually nuclear when it comes to a heat map with pig density. And so we certainly did not want to get into that area. And the remaining concentration of pigs are located in the northwest part of the state that adjoins to the panhandle. If you look at this map inset, you can see Stillwater. The closest swine to our location, which is the star, is actually the small university swine herd there within the Stillwater city limits. And so we selected a site that was university-owned property, so we could maintain control of that. And I will tell you up front that we had to look at the soil composition of probably 3 or 4 other sites before we found one that was the most suitable or ideally suited for above-ground burial, and that's something we'll talk about during this presentation. But that is the site of the above-ground burial process, the star there. So you can see I-35 on the left, and then another highway that goes into Stillwater. So this is what our study site looked like before we bush hogged it and began our study. So it was a site that had previously been cleared a few years earlier and had subsequently regrown in weeds. And so what I consider weeds, it was a mixture of like Lespedeza and milkweed and some Johnson grass and some other things in there. We used a geoprobe, you can see on the right, to to check the soil conditions of this site as well as others prior to initiating the study. These are examples of core soil samples at various depths. We determined the soil composition, it was evaluated, and we also submitted those samples to the Oklahoma State University Soil, Water, and Forage Analytical Lab, SWAPL, so that they could do soil analysis for various components and establish a baseline before we started the study. We also took soil samples at that time for coliform testing because environmental impact is something we'll be looking at in terms of chem and bio. Very wordy slide here. This is the soil profile description. I included that so that people who are interested and knowledgeable on this subject can look at it and read it and digest it. I'm not one of those people, and so this is for others out there that do specialize in soil, and I'm happy that they're on our team. We also utilized insect traps, and so we had, on the left, you see malaise-type traps. It's a net for flying insects, and then we have several pitfall traps for crawling insects. And on the right, you can see the map of the trap placement. And you will see that we have two trenches with carcasses, and so we placed both types of traps immediately around the enclosure, the fence that was around each of these burial sites. We also placed traps about 50 meters away to serve as control so that we could sample the normal insect activity at that site. We were fortunate that that very site, about a quarter mile away, had a mesonet weather data collection system. And so you can see that every 5 minutes or 15 or 30 minutes, a large volume of weather data was collected and recorded and will be available for us to use as reference. In terms of the trenches, we bush hogged the site that you were just looking at. The lush weeds were bush hogged, measured it off, and designated an east trench and a west trench. And as you'll see, we divided the 100 sows into two groups of 50, and that was a logistical decision so that we could accommodate 50 sows and work them up in a single day. And so the study had a staggered start phase of 1 week apart, and we'll talk about that. These trenches were 6 feet wide, approximately 115 feet long, and this is the beginning of where we were marking the trenches. You can see on the left, here it is sprayed off, and we have a backhoe ready to begin digging. And then on the right, you can see 1 or 2 technicians that are refining the trench once it's dug. The trench was approximately 27 inches deep, so about 2 foot deep. And on the left, you can see we have packages of compressed wood chips, our carbon source. So the wood chips were then spread to create about a 12-inch base in the trench. And it was a hot day. This is September in Oklahoma, and I was mentioning to Liz earlier that was probably one of the toughest parts about this, especially for me as I get old, I get soft, and you know, when it's in the mid-90s or high 90s, and we're in this little valley where there's not much wind, it was a hot day. So in terms of our study animals, the culled sows were scheduled and delivered in the wee hours of the morning so that they would be transported from the Panhandle area of Oklahoma in the nighttime, so it would be a bit cooler on the sows. We placed them in a climate-controlled environment, and then they were euthanized by captive bolt euthanasia, followed by use of a stainless steel probe to pith the brainstem of each sow. They were transported out to the burial site in a box truck. So on the right as well as on the top left, you can see the sows in the back of the box truck. Unloaded them onto a prep station that we prepared, and we lined it with plastic. One of our goals here was to minimize any blood contamination of the premises other than the trench so that we could try to avoid attraction of predators or anything else to this site. So the animals were prepped here, We utilized either a skid steer or a backhoe to lift up the sows, and we lifted them by their forelimbs to try to avoid any damage to the femurs, or the hind limbs. Our viral stock, we had those aliquoted into the syringes here and maintained on ice and in shade until they were used. And so we had 10 mil of the viral stock with a tissue culture infective dose 50 of 10 to the 6.8 per mil. And as I said, we kept this shielded from the sunlight and chilled until use. So each of these syringes has 10 mil in it and would be injected into the femur. So the actual process was to place the sow in the trench, and then we would have a team that would reflect the hind limbs back. We would incise on the medial aspect to exteriorize the femur. It was a fairly quick process depending on the technician and the experience of the technician, but it was anywhere from 30 seconds to probably 3 or 4 minutes to exteriorize the femur. We would utilize a 20-volt drill with a 3/4 inch bit to drill into the mid-shaft of the femur. And once we did that, we would exteriorize and replace approximately 10 mil volume of bone marrow. And for the spent sows, the bone marrow is pretty much just one big fatty core. And we discovered in some of our prep work that if you attempted to simply force a volume of like 10 mils into a post-mortem femur, what you end up with is tremendous resistance. It tries to come back out either the hole you're using or one of the nutrient arteries. So we learned early on in our prep work that we needed a mechanism that would allow us to safely transfer 10 mils of viral stock into the marrow and avoid any or minimize any leakage so that we could satisfy the requirements of our Institutional Biosafety Committee. So we would scoop out with a stainless steel spatula about 10 mils of the fat, inject in our viral stock, and then we would use the stainless steel plugs you see on the left. We would take them out of the bag, put a black washer, a rubber washer around the base of it, and then use an electric drill to seed this plug into the femur. Once we had both femurs plugged, and you see that here designated with the arrows on the left image, we would take large zip ties and wrap around the femur because you have to remember we have to go in and collect these. So we'll have to exhume at least the femurs so that we can collect these for testing. And so we use these zip ties to designate right versus left and also give us a little bit of a cord that we could identify as we start to remove the soil during the exhumation process. If you look at the image on the left, you can see one of these basic DeWalt drills that we put the impact socket on just to inject the plugs into the femurs. So this is what the trench looks like with 50 sows placed on the wood chips. Before we covered the sows up, we went through each sow and we incised the abdomen. You can see that on the image on the right. So the abdominal cavities were incised, and that served two purposes. One was to help reduce bloat, because previous studies that Gary had performed demonstrated that when, I believe it was cows bloated significantly, they would elevate the overlying soil and create cracks and fissures. And so we were hoping to minimize that a bit. But the other value of incising the abdomen is that we were able to place temperature probes in the abdomen as well as on top of the abdomen. So this is an example of the hobo data loggers that we used. And on the right, you can see the cords that are either running within the abdominal cavity or on top of the sow carcass before it was covered with soil. And this would allow us to monitor temperatures 24/7 within the carcass and within the trench. Once we placed the hobo data loggers in there, we covered the sows with the soil that came out of the trench to begin with. We also utilized wooden stakes to help us designate each series of 5 sows, because we will be recovering the femurs in groups of 5 sows. So we'll recover 10 femurs at each collection interval, and this would help us to keep a visual idea of the segregation of each group of 5 sows. So here's the final measurements of the trenches. So we utilized 50 sows per trench. The sows averaged about 450 pounds each. The west trench was 6 foot wide and 113 feet long, and the east trench was just slightly bigger at 6.5 feet wide and 117 feet long. Once they were covered, you can see a graduate student here spreading seed, ryegrass seed, because it was fall when we did this study. We have it fenced off to minimize any predation. You can also appreciate the stakes, and these are, as I said, at series of 5 sow intervals. So this is what the site looked like on September 12th of last year, the day that we finished phase 2 of this staggered start. You can see in front of the white truck on the left is our control trap for flying insects, and it's a little bit harder to see the other traps in this image, but I will show them to you. So this is what each trench looks like from the end, and I think here, if you look on the image on the right, you can see black structures on each side. Those are the traps for the crawling insects, and then about halfway down on the right image, you can see a malaise trap for flying insects. If you look at the dates here, on the left is the day that we finished phase 2, and on the right is not quite a month later with the ryegrass seed growing over the top of the burial site. Motion detectors did pick up activity. Most of the activity was outside of the fence, but not all of it. You can see on the left there's a deer. There were a lot of deer out there. We did not observe feral pigs, to my knowledge, even though Oklahoma has a lot of feral pigs. On the right, I believe that's a possum that is in there, and we also saw occasional birds like a turkey vulture or turkey, but there was no significant or detectable disturbance in the soil, so we didn't have evidence that things were getting in there trying to dig into the carcasses. So if we move on now to the actual results that we have on hand that have been QC'd, we have through the first 6 months solid data, and so this is the east trench, and what we're looking at here is virus isolation. So we had 2 main objectives in terms of the viral component. One, could we detect infectious or live viable virus? And the second is, could we just detect viral DNA? So we had those 2 questions to answer. And this is the end of week 1, so day 7. And in this east trench, you can see that we recovered viable virus from the femurs of 4 of the 5 sows. 1 sow, we did not recover viable virus from either femur. The others we recovered from all. We have the PCR CT values on the right, and you can see that they range from 22 up to 38. This is the west trench at the same collection period, so day 7. And on the west trench, we recovered viable virus, potentially infectious virus, from 3 of the 5 carcasses. And again, they all had pretty hot CT values. And femurs were harvested at the ends of weeks 1 through 4 and months 2 through 6. At least that's what we have data available right now. And viable swine pox virus was only recovered from the femoral bone marrow at the end of week 1. So at the end of week 2, we did not recover viable virus. And even at the end of week 1, the bone marrow sample had a low titer of 100 virions per mil. As I said, no viable virus was recovered from the end of week 2 through month 6. And that demonstrated that the surrogate virus, swine pox virus, was rapidly inactivated between day 7 and day 14 in the femoral bone marrow of these cells. In terms of PCR, not surprisingly, we were able to detect viral DNA from all carcasses at all time points through 6 months. And that's not really a surprise. We anticipated that type of finding given that the site was fairly privileged, and even though the virus was inactivated, we still expected to see that. And the CT values at the end of month 6 were still fairly hot and ranged from 24 to 33. At the end of the 6 months, so on the 6-month collection, only a single femur from one carcass had an undetectable PCR assay. In terms of insects, lots of insects. So extremely diverse and extremely abundant. So 6,339 crawling insects have been identified at least through August 20th, as well as 10,140 flying insects. Most of the insects that we sampled were true flies. Overall, and again, this has not been applied to statistical analysis, but no major differences were identified in the control traps versus the traps that were adjacent to the burial sites. But again, very preliminary data, and I suspect that they probably have 1,000 or 2,000 more insects to add to this data set by the middle of September. So our conclusions is that under the conditions of this study, and this study being placed where it was in Oklahoma, swine pox, the surrogate virus inoculated into the postmortem femoral bone marrow of cull sows, was rapidly inactivated in the first 2 weeks or prior to week 2 of above ground burial decomposition. Swine pox viral DNA was detectable in virtually all samples from all carcasses through the first 6 months, as expected. And importantly, the detailed analysis of the study data is certainly underway, and we hope to have more to share very soon. I want to acknowledge all the team members. There were a lot of moving parts here. Certainly Lori Miller, who did a phenomenal job in terms of being a study coordinator and communicator and keeping us on task and keeping us moving forward. Gary Flory for his technical expertise. It's my understanding this is probably his 3rd or 4th species to do this on. I want to recognize the National Pork Board again for generously providing the sows for us to use, and then of course USDA for the funding to do this project. And I'm happy to defer back to Liz for questions, and I'll try my best to answer. I do have my email on the first and last slide, so please reach out to me if I can't answer your question or if you just want to chat offline, and I will point you in the right direction.
We do have a couple questions coming through the chat. Any preliminary data or comment on trench temperature kinetics?
Yes, a lot of data. Unfortunately, I don't have that available. If you will send me an email, I'm happy to send you what information you would like. I can show you the first week. This is the reading from the 11th of September through the 22nd of October 2019, and there are a lot of variables here. The biggest up and down line, I think, is the environmental temperature, I believe, because I think that was some night temperatures that were up and down. I think the blue bars are rain, and shortly after starting this study, we received a pretty decent rain, and so you know the trenches were saturated. There's no doubt about that, but we do have this data, and I'm happy to at least share what we can if you will just let me know your email address.
So what I can do, Keith, is I can send you the email address for the person who made that question. I'll make sure I get that to you. Yeah.
Okay.
How did you determine that the virus was viable?
Yeah, so for viability, it was just virus isolation. So it was cell culture. And it's not difficult for people, you know, for virologists to grow swine pox virus. In this case, it was Fernando. And the challenge was getting a really, really high titer because swine pox virus is typically does not grow to an extremely high titer. So that was probably one of our bigger challenges than just simply utilizing cell culture to look for live virus.
Okay. Will the time necessary for completing the whole composting process be evaluated as well?
Absolutely, yeah. And so I haven't seen data from this past month in terms of decomposition of the bodies, but that's one thing we want to ensure and document is the stage of decomposition, the degree of decomposition prior to land reclamation. So we're kind of in that phase right now, and I can certainly get more information on that, but I don't know that information right now.
Okay. What soil characteristics are conducive for above-ground burial, erosion, leachate concerns?
Yeah, that's a great question. As I said, we had to sample, I would say, at least 3 or 4, maybe 5 sites. So just within that 5-mile perimeter of Stillwater, I believe we had one site that we hit bedrock, like 2 or 3 feet below the ground, and another one I believe we hit water, very, very shallow. And so that's one of the main considerations for this application is does the farm, does the site or premise have soil that's conducive to this accelerated aerobic decomposition process? And quite frankly, probably a lot of them will not. But what we're hoping to do is just put this technique into the arsenal of options that premises will have. And ideally, what they could do is just have soil evaluated, and they will know when they formulate their disaster plan of whether or not this is an option on that premise. But as I said, I would have to defer to Jeremy Seeger, who previously was at the Oklahoma Department of Ag, because that's his specialty is looking at soil parameters and their properties that would be conducive to this. But I don't honestly have the answer to that. Gary Flory would certainly know. Jeremy Seeger would be a good reference as well.
Did you perform any leachate measurement?
We did early on, and primarily because I mentioned the rain, and you can see the bars here. After the first large rain, and it was, I think, in the magnitude of 3 or 4 inches, there was literally visible water standing on some of these trenches. And not only was visible water, but also blood. There was some blood in the water because it was in the first week. And so we collected those, and 2 things we did were coliform counts and PCR to see if there was any virus that could be detected. And so in this case, we were looking at the biological components and not necessarily the environmental degradation components. In that case, we did recover a lot of coliforms from that water. We did not detect any virus by PCR in the water that was visibly contaminated with blood.
Were the insect traps tested for presence of viral DNA?
No, we didn't have that as an endpoint, and so very good question. And I say that because, as you know, certain insects have been implicated in transmission or potential transmission of African swine fever. And so what we were hoping to do is to at least characterize in our region what type of insect activity may be increased. But that's a very good question, and certainly something that would have to be looked at closer if someone was using a different surrogate virus for sure.
Okay. Did you monitor near the trench for potential leachate, and if you did, was that analyzed for viable virus?
Just as I mentioned earlier, we looked at the leachate by PCR, and we stopped there because clearly if we can't detect any viral particles or viral DNA by PCR, there's not a chance that we will have virus that's viable there.
So do you feel that showing no virus viability but PCR positive results will be acceptable by VS to show proper disposal has taken place?
I think so. I think it's a very compelling argument. It's an argument that we being the U.S. have used on other instances. For example, PEDV. There's certainly in studies you can detect PED in various substrates, but certainly not a viable virus. And so I think I think it's fair, but a lot of it depends on the methodology and how much you trust the methodology.
Was the soil reevaluated regarding biota, and if so, have you determined soil property variables relevant to viral retention or destruction?
Yeah, that's a really good question as well. We really did not look at the whole microbiota. We were looking specifically at coliform counts and the environmental impact, but you know, I think that would be a fascinating study to do, and we now have sophisticated tools. And they're becoming more user-friendly and cheaper to where we could do whole genome analysis on some of these samples. But it was not part of this study design. Our basic concern was there a risk of contaminating the water table? So that's really our big concern there. And if so, you know, could you monitor it by something pretty simple like coliform counts?
Any unexpected observations or issues that need to be addressed in any future studies?
I'm sure there are. You know, one that we ran into pretty early on, and this was a design issue, is that we had intended to establish baseline for soil bacteria. However, we utilized the geoprobe to collect those, and during the collection process, the samples were primarily processed for soil composition analysis, with the coliform counts being a secondary concern. And of course, in that process, we very quickly discovered that you know, the more sensitive indicator in that case would have probably been testing for bacteria before you know, soil samplers were ground up, and so we know we got cross-contamination there, and the baseline soil data is certainly not reliable for that reason. And so that's one thing we learned, but I have no doubt that as we flesh through the rest of this data, we'll come up with you know, other gems that can help us plan a better study next time.
So did you evaluate any spillover contamination to the underground soil?
That's what we're doing now. And so the samples that we collected from the baseline were just to see if this would be a good site to use as well as establish our baseline. During the study, the samples were literally collected from the trench. And so we collected those samples, and they're currently being analyzed. So that is our primary concern, is there vertical or lateral transmission of anything that might impact the environment.
Any monitoring of constituent movement through the soil under the trenches?
Just what I just mentioned, basically, but that would be essentially looking at soil composition and changes in the descriptive properties as well as coliform count. So that's pretty much the extent of our soil evaluation, but it goes, I think, 10 or 12 feet or maybe 16 feet below the trench.
Okay, we have a request to show the soil slide again.
Oh, sure. Yeah, hopefully there's someone there that can help me understand some of this.
So we'll go ahead to go to the next question. What is the time recommendation for putting the ground back into its normal use?
I think that's going to depend on the decomposition process. In this case, this was university property that was not used. If you think about a farm setting or infected premise setting, you know, really the primary concerns up front are going to be addressing the issue, so controlling, you know, biocontaining the virus, providing biosecurity by keeping it on the farm, and then farm sustainability. You know, how quickly can the farm return back to operations? You know, the other options would be deep burial, in which case we know that the animals are likely to be entombed for months, if not years, and then without that, and incineration on site. If you get an incinerator on site, your options get few, and they may have to, carcasses may have to leave the farm at that point, which certainly poses a biosecurity risk.
Please describe why the researchers chose 50 sows. Was the sample size analysis performed, and if yes, what is the statistical power of the data?
Yeah, we discussed that at least on a couple, 3 meetings, and you'll notice a couple of our co-authors are statisticians. And so we asked them to model, and I honestly don't recall all the specifics of that discussion. And so, you know, we were looking at various endpoints and parameters, but quite frankly, not being a statistician, what I typically do on my studies is rely on their input to help determine what is the lowest number of animals that we can use to still show statistical significance. And so I can say, yes, we had statisticians involved. Yes, we utilized their input. But unfortunately, I'm not well-versed enough to say more than that.
And there's a thank you here to the entire research group to address one of the crucial issues in foreign animal disease control techniques. So, and I have another person that's interested in trench temperature monitoring information, and I will send you his email address. Is there a desired end product with above ground burial, or is the intent to leave it on the landscape?
Well, I'm not sure what they mean by leave it. Do you know, Liz?
I don't have anything else. Maybe something will come through in the meantime.
Yeah, because the goal is to have complete decomposition of animals so that nothing remains there. For us, we'll have things to collect as we do reclamation, so we'll have all the wires and things to collect at our site. Take the fencing down. I mean, we have to do things for regular reclamation.
Okay. Do you think that the Kinetics swine pox inactivation in femur is representative of other tissue and organs?
Yeah, that's probably the biggest question here, and the answer is I don't know. Our approach was to try to determine if you had a tissue that was the most privileged site and probably the least directly impacted with a very active decomposition and the aerobic decomposition, what would be the longest it would survive? And so we were looking at it from that standpoint. But it's a very fair question. There are other researchers who are looking at different viruses in decomposing swine. And so I think as more and more information begins to be peer-reviewed and disseminated, I think folks will be able to build on that and design some really nice studies that hopefully, even using ASF will be able to answer that question. But that's a very good question, and we don't know the answer to that. But I certainly look forward to comparing our data to, for example, other types of viruses. We just tried our best to pick a virus that would closely mimic it and put it in a privileged site so that we could see how long it might be viable. That's kind of what we were going for.
Okay, were there any soil samples collected underneath the trenches after or during the study? And what does the project site look like now?
That's a good question. I'm in Illinois now, and so it's not like I can drive by there, but I did talk with Fernando and collecting the last femurs, because we had to go and make sure we collected all the femurs. But he made it sound like that there was advanced decomposition in the trenches because they had to exhume those, at least the rear ends of the sows to collect those femurs. And again, it was September, so it was hot, and it didn't sound like it was the most pleasant job, quite frankly. So that's something we'll monitor, and in all likelihood, if there's not adequate decomposition or complete decomposition in the 12 months, I'm quite confident that we will extend this to where we're monitoring so that we can have a complete data set.
The next question is, when you say 12 inches of wood chips under the carcass, do you look for that 12 inches before placing the carcass on the bark, bed, or after? Just wondering if you see the carcass compress down the wood chips considerably.
Yeah, good question. There's no doubt, just walking on the wood chips, they will compress down to where they're maybe 2 or 3 inches or 4 inches deep. Certainly putting these 450-pound sows on there, it gets compressed down. But it's still a rich carbon source, and we're not looking for it to necessarily be super fluffy to increase the aerobic decomposition, even though for those who have been in the decomposition houses they use for swine, where they pump in oxygen underneath the carcasses, if it is looser, it will certainly help with the aeration, but really we're after the carbon source there.
So at the end of your study, was the compost feasible to be used as fertilizer for agricultural use?
Ooh. Yeah, I don't know the answer to that. I suspect, and I'm guessing here, based on the end of month 12 collection of femurs, that everything was pretty messy and probably hard to tell that it was distinct wood chips would be my guess, but I'm speculating.
What was the end point for sufficient composting of the carcasses? 3 months, 6 months, 1 year, or ongoing?
I would say ongoing. At least that's my understanding from folks that are on site and that did the 12-month recovery. I may stand corrected, but that's my understanding. I know that we had femurs we could recover that were not a challenge to recover, so I'm assuming that it's still ongoing. But that's a good question, and hopefully we'll follow it out until at least Gary and his team, who are certainly more authorities on this, consider it at a satisfactory point.
Okay. Any perspectives or info from other countries that may have tried above-ground burial as a disposal method for an actual ASF incident?
I don't have direct communications on that. I have heard indirectly that there have been talks in other countries that are combating ASF, like Vietnam right now, to try different strategies, but I would literally have to defer to Gary because he would be involved with that if that was the case.
Is there any plan to introduce sentinel pigs in the site tested after a certain time?
No. I think we're pretty confident, based on our sampling and our testing, as well as our design, that if some swine pox was left behind, it was very minuscule quantities. And the reason I say that is, we typically think of bone marrow, like the femur, of being a closed system. But when you have nutrient arteries at each end, there is potential for leakage. And so I can't say it's absolute, but I feel that we recovered certainly as much as was feasibly possible to recover. But would I be surprised if feral pigs out there were dealing with swine pox, anywhere in the state? No, I wouldn't be surprised at all.
Okay. So Gary Flory wrote in and said, based on the Oklahoma and previous studies, we have generally observed that the site can be returned to previous use.
Thanks, Gary. Appreciate that.
Okay. We have someone that's interested in internal temperature monitored during the study. They're asking, will the slides be available on the VS website? This is being recorded, and that will be available. How much lead time was needed and required for this excellent study to start?
I believe we began our first discussions around maybe February of 19, and that was just getting some players together and identifying the players. And then we had to flesh out, we met, I believe, as maybe every other week, had to flesh out what were the primary objectives. The study grew a bit as we identified other areas of expertise there locally. And we had planned to start in June or July. That was originally our plan, but identifying suitable soil properties and a site posed a challenge to us because, as I said, we tested a few and they just were not considered adequate or ideal. We weren't going for the best case scenario, but we definitely wanted to have the best chance of success.
Okay. There's a couple more questions because we're nearing the top of the hour. The one thing I will say for people that are interested in other data, I'll make sure that I get this information to Dr. Bailey, and we'll make sure that I send all of the people that are on your first slide, if I can get their emails, we'll send it out to the participants. The next question is how much might soil properties affect decomposition and viral activity under the trenches?
If I understood the question correctly, you're talking about the microbial activity?
Yeah, the soil for this site is quite high pH, perhaps higher than most areas where this method may be used.
Okay. Yeah, that's certainly something to consider because we're relying heavily on the microbial activity to advance the decomposition. But what I don't know, if you think about this, the carcasses are on the carbon source primarily, and so what I don't know is how much the soil would directly impact, and to what extent they would impact, the carcass pH and decomposition process. I don't have an answer for that.
Okay, and Gary Flory also wrote in and said at year 1, we observed excellent decomposition and the site was graded and seeded, returned to previous use. And I think there was another question regarding was it used in other countries, and Gary wrote it was used successfully in Tunisia for FMD and blue tongue, but I don't think it has been used yet for ASF.
Great, thanks Gary, appreciate that.
There's another question. I'd be interested in knowing if the carcass is froze at any point in time.
I don't know the answer to that. I suspect it did, and we certainly have the hobo data loggers to tell us the temperature inside the carcass. Typically, winters are pretty mild in Oklahoma, and so I would say much less likely there than, say, for example, South Dakota.
Okay, let's see. Will there be a final report that presents all of the data with conclusions?
Absolutely, yeah. So there's multiple facets of the data here, and so it'll probably be presented in terms of the biological activity, the adequate nature of the above-ground decomposition, the insect activity and abundance. So it'll be hopefully in the public domain in the next few months as we can look at detailed analysis and statistical analysis on the data. But absolutely, we want to get this in the public domain as soon as possible, and that's one of the reasons we're sharing it here as well as at the AAV meeting, just so that even though it's extremely preliminary, people can see the data that we have, and as they start designing studies, integrate some of what we learned into their study.
Okay, and the last question I have is, have there been any study to compare the virus activation efficiency and efficacy between regular composting and above-ground burial for ASF, I think they meant?
I don't know the answer to that. I doubt specific for ASF, but you could certainly look at other microbial endpoints. That's not necessarily ASF virus. And I'm sure that has been done to some extent, just because the swine industry is embracing composting.
Okay. Well, this is great. A lot of great questions. So I thank you, Dr. Bailey, for your presentation and for all those answers. I will make sure that you get the chat questions. I'll email them to you once I receive them so you can see the information. And I just want to thank everybody who attended today, and I'll be sending out more announcements for future webinars, and so please watch your email, and hopefully we'll join again. So have a great rest of your day.
Thank you, Liz. Appreciate it.